introductions. So I'll make the presentation very brief. If require, we'll spend more time in discussion. Okay. Yeah. So as we are talking about drug designing, first we must know what is a disease. Okay. Disease is any harmful deviation from the normal structural or functional state of an organism, generally associated with certain signs and symptoms. Like deviation from the normal physiological condition, harmful deviation from the normal physiological condition of the body. It may be due to some external factors, it may be due to some internal factors. So any such deviation is known as a disease. And a drug is a chemical substance, typically of known structure, which, when administered to a living organism, produces a biological effect. That means when we suffer from some disease, the normal physiological condition of the body is disturbed or deviated. Now, drug try to restore the normal physiological condition of the body from the deviation. A drug is also called a medication or medicine and is used to treat, cure, prevent, or diagnose a disease or to promote well-being. Now, these are very basic definitions of disease and a drug. There are several other definitions you will find in the literature. Now, since my topic is about disease target and molecular interaction, the concept of disease target or drug target is not very new. Like we know the Ayurvedic formulations, okay? There we may not know the active principle or the active molecules which are responsible for the therapeutic uh, property, but almost 100 years back, Paul Ehrlich, okay, he was a German scientist, and you know the homeopathic, the origin of homeopathy. So Paul Ehrlich, he believed that whatever drug we take, okay, it ultimately targets to a particular molecular entity inside our body. It may be a protein, it may be a DNA, it may be an RNA molecule. So, or it may be a molecule. So Paul Ehrlich, he believed that the drug molecule, it termed the drug molecule as the magic bullet. And these magic bullets, it's like we are hitting the bullseye, okay? So we are hitting a particular protein inside the body. So the drug molecule or the magic bullet always binds to the drug target or the bullseye. Now, Professor Prabodh Bora sir has already given the overview of drug designing process. So the first step is always target identification. We must know which biological entity we are targeting, okay? And after target identification, there may be several approaches like ligand docking. In the second part of my lecture, I'll be discussing how a ligand or small molecule binds to the target molecule, which is a macromolecule. So this is studied using the ligand docking approach. Then we perform homology modeling when we don't know the structure of the target. Then there are some other approaches like QMMM, quantum mechanics, molecular mechanics, hybrid approach. Then fragment-based drug designing, where when we know the structure of the target protein, and if we can identify the active site, we can design de novo synthesize a small molecule which fits into the active site of the molecule. Then we can go for molecular dynamics simulation to study the stability of such interactions. So Mr. Nobojyoti Goswami is going to discuss MD simulation after my lecture. Then this is the main approach. So in this particular presentation, my focus will be on target identification as well as the molecular interaction. Now, what is a drug target? A drug target is anything within a living organism to which some other entity like a ligand or a drug is directed and or binds, resulting in a change in a desirable therapeutic effect. Like here we can see, we know in our body there are different enzymes, okay? They perform different vital functions inside our body. And these enzymes have got their native substrates, okay? Like suppose cell division is a process, and you know during cell division, the microtubule formation, for the formation of the spindle fiber, the tubulin, alpha tubulin unit must bind to the beta tubulin unit to form the tubulin heterodimer. Okay, so you can say that tubulin A or alpha tubulin and beta tubulin, they bind to each other in the normal condition and cell divides. Okay, so if this is alpha tubulin, you can say this is a beta tubulin. Or I'll just reverse the name. Suppose if this is beta tubulin, this is alpha tubulin. Okay. Now, if you want to design a drug against cancer, what you can do, you can try to think to inhibit that process so that the heterodimer formation does not occur. So what you can do, you can design or you can identify one inhibitor which fits into the active site of, suppose, the beta tubulin. As a result, the alpha tubulin cannot bind there anymore. Like the colchicine. Okay, colchicine is a known inhibitor which disrupts the heterodimer formation in microtubule. Okay, so suppose this is colchicine, it binds to the active site of one of these tubulin units and it inhibits the formation of this heterodimer. As a result, the cell division process can be checked. Okay, and in this case, this is known as a competitive inhibitor, and there are non-competitive inhibitor also. Like it directly does not bind to the active site of the protein. Instead, it binds to some other place and distorts the shape of the active site so that the substrate cannot bind. So, while in drug designing, the first step is to identify that crucial protein or receptor or the portion of DNA or RNA in our body or inside the pathogen, by blocking which we can think of curing the disease. Okay, and here I am talking about blocking. There are some drugs which promotes the activity of the enzyme. Okay, there are two kinds of drugs, agonist and antagonist, and most drugs are antagonists. Okay, they inhibit the activity of the drug target. Now, depending on the diseases, Borasar also showed this thing. Depending on the type of disease, pathogenic disease and non-pathogenic disease, drug targets may be different. For pathogenic disease like malaria, tuberculosis, cholera, or this COVID-19, typhoid, leishmaniasis, rabies, etc., we may target a vital protein from the pathogen so that if we block the activity of that particular protein or enzyme in the pathogen cannot survive or it cannot replicate. Okay, like here I have given some examples like the inside pathogen we may think of enzymes associated with cell wall synthesis, we may think of enzymes associated with protein synthesis, we can think of enzymes associated with nucleic acid replication and transcription of the pathogen. Okay, or inhibition of essential metabolite synthesis. So we can think of targeting such essential enzymes or proteins or receptors in the pathogen. while selecting drug target, or in such case, you must be careful that because there may be a, whatever target will select, there should not be any similar, highly similar protein in the host's body, in the human body. Okay, otherwise it will inhibit that that human enzyme also, and which may cause some adverse effect. And in case of non-pathogenic disease, since no pathogen is involved with the disease mechanism, we are bound to select one human native protein itself, like diabetes, most of the cancers, cardiovascular disease, Alzheimer's, etc. So in this type of diseases, there is no pathogen involved. And since no pathogen is involved, we are compelled to select one of our own protein as the target protein. Like I have cited the example of alpha tubulin, beta tubulin in case of cancer. Okay, beta tubulin is a drug target for the popular cancer drug Taxol. Now, in this presentation, I'll be mainly discussing what is a target and how we can identify the target, then the ligand or the small molecule. See, I'm using the term ligand because I cannot use the term drug initially. A ligand, when it passes through some test, it shows good result in some experiments. When it passes clinical trials, then only I can use the term drug. Otherwise, I cannot use the term drug. So drug is a ligand, okay, but all ligands may not be drug. So our aim is to identify the ligand out of several thousands or lakhs of ligands, which ligand may act as a drug. Okay, remember one thing, the ligand always interacts with the target molecule, and that interaction is also very important, and that we study using the molecular docking approach. So first step is target identification, then we take a set of ligands, then we try to find out how these ligands interact with the target protein. Now, what is a good target? Identifying a biological target that is druggable. Target is termed druggable if its activity can be modulated by therapeutic, whether it be a small molecule, drug, or biologic. So, as I have already said, that a protein or an enzyme or a receptor or a portion of DNA or RNA can be termed as the drug target only if we can modify its activity, okay, by fitting some small molecule or some macromolecule to that particular target, and which results in some therapeutic effect, then only we can call it a druggable target. And I have already told you that target should not have any highly similar protein in the host's body. Otherwise, it will cause some adverse effects in the host. And this statistics, we can see the GPCRs, G protein coupled receptors, the most of the drug targets, almost 13%, are GPCRs. Then the different enzymes, kinases, proteases are also different drug targets. Now, the most important step in the drug discovery process is the target identification. Okay, it's like fishing the targets from thousands of proteins inside our body, and our aim is to find out which protein may be selected as the best target for a particular disease. Okay, so there are different approaches for drug target identification. Target identification and characterization begins with identifying the function of a possible therapeutic target, gene or protein, and its role in the disease. Identification of the target is followed by characterization of the molecular mechanisms addressed by the target. Now see, there are several databases available. These are some of the several other databases, okay, several databases available in the internet. Like there are different databases like KEGG pathway databases, there are human metabolome databases there, from where we can see different metabolic pathways. We can see the graphical representation of these pathways, and from these pathways we can identify the crucial proteins or enzymes involved in this metabolic pathway, so that we can identify which enzyme may be target to block a particular pathway. Like for cell division, what are the enzymes and proteins involved in the cell division process? If you are to design a drug against cancer, then see the cell division pathway and see the different enzymes and proteins involved, and try to identify which protein may be selected as the drug target. Now, once you identify a drug target, you can use some functional screening to see whether it will act as a drug target or not. Like you can knock down that particular gene or knock out that particular gene in some model organism and see whether the disease is getting cured or not. Then that you can do in experiment purpose, but that will tell you that if you block the activity of that particular protein or enzyme, the disease may be cured. So we can screen that functionally in wet lab. But in dry lab, what we can do? See, here, now see, in the drug designing thing, we cannot knock out or knock down a particular gene from the patient. So instead, we try to identify a ligand or an inhibitor for that particular target protein, so that the whatever knockdown or knockout we did in the screening, at the time of screening, we can do it by using some inhibitor molecule. Now, in the dry lab part, see, suppose you have identified a particular target protein from studying the metabolic pathways. So what you can do, you can go for some gene regulatory network analysis to study the associated genes, okay? To study the associated genes. Just a minute. With the target protein, and just a minute. Can you see this PDF?
Yes, yes, it is visible.
Yeah, this is one of our works where we did gene network analysis of efflux pump proteins in Shigella species. Like Saurav sir also told you that the microbes are getting resistant against different antibiotics. There are different mechanisms, and one of the important mechanism is the expression of efflux pump. So whatever antibiotic you take, it goes inside the bacterial cell, and the bacteria, they develop some pump protein, and this pump protein, they pump out the antibiotic, thereby reducing the concentration of antibiotic inside the bacterial cell. So antibiotics fail to act against the bacteria. Now here in this particular paper, we tried to find out the different associated genes with our target protein ACRB. Sometimes it may not be possible for us to directly block the activity of ACRB protein. Suppose ACRB is a particular target protein, and sometimes due to different regions, it may not be possible for us to find a suitable inhibitor for ACRB. Now we may think of other associated genes or proteins, okay? Other associated genes or proteins which may be inhibited. So from this network also we can think of targeting the other proteins. So in this way also we can identify the drug target. Now I think I am again back to the PPT. Now another example of the drug target is, suppose this plasmapsin 2 as a drug target for malaria. Plasmapsin 2, which is present in the food vacuole of Plasmodium falciparum, the causal organism of the cerebral malaria, it hydrolyzes the bonds linking certain hydrophobic residues or amino acids in hemoglobin or globin domain. Okay, you know the Plasmodium parasite, it takes our hemoglobin as its food material, and plasmapsin 2, it breaks down the hemoglobin inside the food vacuole to prepare its food material. Now if we block the activity of this plasmapsin 2, hydrolysis of hemoglobin does not take place and the parasite starves for food, and as a result if we block plasmapsin 2, will be the malaria may be cured actually. So therefore plasmapsin 2 is regarded as an important target for malaria. Okay, now after identifying the target, what is the next approach? Get the 3D structure of the target. So three-dimensional structure is very, very important. Check if it is already available in the 3D structure databases like RCSB Protein Data Bank, and if it is not available, you can think of predicting the structure using some bioinformatics approach like comparative modeling or homology modeling, threading, or some ab initio prediction. So knowing the structure, particularly for computer-aided drug designing, knowing the structure is very important, structure of the target, because otherwise what we will input in the software? We need that structure. Then after getting the structure, okay, next step is find suitable ligands that bind to the target protein. So there are different ligand databases from where we can take the ligand structures, and we call them different ligand libraries. So these ligand libraries, first what we do, we pass the ligands through some physicochemical filters. These are not some physical filters. Actually, these are some physicochemical properties that a molecule will have to fulfill. Otherwise, those molecules will be discarded. These filters are also known as Lipinski filter, Weber filter, Admet filters, etc. So we consider the ligands for further screening only when these molecules pass through these physicochemical filters. And those molecules which pass through these physicochemical filters, we consider them for further screening, and we have suppose we have already identified the drug target, and the structure of the drug target is also there. And the next step is to find out which ligands from these filtered ligands, which ligands interact with the target protein, and if it interacts, to which portion of the target does it interact. Okay, one ligand may interact here, one ligand may interact here, or one ligand may interact to the active site. So which ligand interact to which portion of the drug target, that can be determined by the docking studies. And okay, so the interaction study is another important step for drug designing, and that is performed through molecular docking. Now, the very basic Lipinski rule of 5, Christopher Lipinski, he studied some already available drug in the market, and he tried to find out what is common in those already available drugs. Okay, and he ultimately proposed that for the orally administered drugs, the drugs are better absorbed and permeate if the molecular mass of the drug is less than 500. Okay, and the C log P value, see, molecular mass indicates the complexity of the molecule. If it is the simplest measure to uh, determine the complexity of a molecule. If the molecular weight is very high, the structure will also be very complex, and it is always believed that simplest is the best. Okay, so 500 is considered as the optimum. There are some exceptions, like Taxol molecular weight is around 900, I think. Then low lipophilicity, it should not be very highly lipophilic. So the C log P value, it should be less than 5. It is known as the partition coefficient. The value should be less than 5. What happens if the drug is very lipophilic? When you take the drug orally, it will be absorbed by our intestinal tract, the gut, and it will whenever it will try to penetrate the cell membrane, since the cell membrane is made up of lipid bilayer, it will remain inside the cell membrane itself if it is very high lipophilic. Okay, it should be lipophilic, otherwise it will not come in contact with the lipid bilayer, but it should not be very highly lipophilic so that it cannot penetrate and enter the cell and reach the cytoplasm. Then it should not have more than 5 hydrogen bond donors and 10 hydrogen bond acceptors, so that it does not form very strong bonding with the target protein. Now, why it is important? Because there is another filter which stands for ADMET: absorption, distribution, metabolism, excretion, and toxicity. So, as I have already told you, when you take one drug orally, first it must be absorbed by our gut, okay, the mucus layer of the gut. Then after absorbed, it must be distributed evenly throughout our body. Okay, that is the next step, distribution. Then so many high energy metabolic activities, metabolic reactions are going inside our body that our drug may enter in some of these metabolic pathways and it may be degraded. So whatever drug you take, the metabolic pathways may degrade the drug molecule. Then finally the excretion. Drug, they help us in curing the disease, so they are beneficial, but it should not get accumulated inside our body for longer duration, otherwise it will develop toxicity, because it's a foreign substance after all. That is why I have told you it should not form very strong bonding with the target protein. So ultimately it must be excreted out, otherwise it will develop toxicity. And last is the ADMET, the T, it stands for toxicity. So obviously the drug molecule should not have any toxic effect. So using bioinformatics softwares, we can predict all these properties, whether it will be absorbed, whether the drug will be distributed, whether it will be able to bypass the metabolic pathways, whether it will be excreted out or not. These things can be predicted, whether it is having some mutagenic effect, whether it is having some carcinogenic effect, whether it is having some allergenic effect. These things can be tested using some software. Now come to the molecular interaction part. As I have already told you, the drug molecule interacts with the target protein. Now here, suppose B is the drug molecule and A is the target protein. So in molecular docking approach, what we do, we study the binding of these two molecules, and whenever the drug molecule spontaneously bind to the target protein, some amount of energy is released, because it's a concept of thermodynamics. See, in the free state, if this is their energy level, when they bind to each other, their energy level comes down. From this step, the energy level comes down. So, you know, energy can neither be created nor be destroyed. So it is just transformed from one form to the another. So initially it was at this level. So from this level it comes to this level. So this much energy will be released in the form of heat energy. So when the drug binds to the target protein, it forms the complex and ultimately it releases some amount of energy. And from the amount of energy released, we can correlate their affinity, how much affinity is there between the drug and the target protein. And see, I have already told you where does this energy come from. Okay, the energy comes from the hydrogen bond formation between the drug target and the drug molecule, van der Waals interaction, and other weak electrostatic interactions. So the drug binds to the target protein, gives some stability, and you know, when it comes to the lower energy level, it becomes stable. So that much energy will be released in the form of E total. Now in docking, you will find that E total is prefixed with a negative sign. Negative simply indicates the release of energy. And a high E total, high means high negative E total, indicates good binding. And the value E total of ligand enzyme binding must be comparatively more than the enzyme substrate binding. Like I gave you the example of tubulin A and tubulin B. They bind to each other, and suppose minus 20 kilocalorie per mole energy is released. Okay. Now suppose colchicine. Okay, colchicine is a drug. It binds to tubulin B, and this is suppose colchicine, and if colchicine must inhibit the alpha beta binding, then this docking must give you around suppose minus 40 kilocalorie per mole energy. That means the drug must have more affinity towards the target protein than the affinity of the substrate towards the target protein, of the natural substrate towards the target protein. Now, what do molecular docking study? What does molecular docking study tells us? Okay. Does a ligand bind to the target? Yes. It will tell you whether a ligand will bind to the target protein or not. And if it binds, to which position of the target does it bind? How much energy is released upon binding of the ligand to the target? What different kinds of bonds are formed between the ligand and the target? Okay. So from a molecular docking study, we can get the answer for all these questions. Dr. Aurobindo Ghosh asked one question about flexible docking and rigid docking. Whatever structures are available in RCSB PDB database, most structures are determined using X-ray crystallography, where the protein is first purified, then it is crystallized, then single crystal, through the single crystal X-ray is passed through. And by studying the diffraction pattern of the X-ray, the structure is determined. So the structure which we get, that is a crystal structure. Now, the problem is inside our body, the protein is not in crystal form. So it is not a rigid form. It has a dynamic structure, okay? And the ligand also, it has got several rotatable bonds. So we must specify the rotatable bonds. There are some softwares like you can perform flexible docking with Autodock 4.2 also, where you can specify some of the amino acids as flexible, and you can specify the ligand also as flexible, so that you get a more accurate result. So thus, from a library of compounds, best compound or best inhibitor may be predicted. The stability of the interaction can be studied by molecular dynamic simulation, which Navajyoti Goswami is going to discuss after my lecture is over. Then, before concluding, I'll show another paper. Yeah, so this is a paper where we all are involved, like Dr. Saurabh Mohanto sir is there, Professor Bhaven Tati sir is there, Professor Prabodh Bora sir is there, Navajyoti Goswami is there, Professor Pratap Jyoti Handik, Honorable Vice Chancellor of Guwahati University is also there, where we tried to identify the potential plant-based inhibitor against the viral proteases of SARS-CoV-2. Professor Prabodh Bora sir mentioned two important proteases in the morning session, which are responsible for cleavage of those viral polypeptides, so that the functional proteins can be formed. And why I am showing this paper, because here you can see we took these proteases as the target protein, and we took a library of compounds, ligands, and we performed the docking experiment when we dock a huge number of ligands with a target protein, that is also known as virtual high throughput screening. And we tried to find out the best ligand, best inhibitor. Then we did the docking, then we studied the stability of that ligand target complex through molecular dynamic simulation. But why I am showing you this paper here, because the Navajyoti Goswami told me to mention few things about QSAR, quantitative structure activity relationship. In this particular paper, I was not involved in docking or simulation studies. I was involved in the QSAR study. QSAR stands for quantitative structure activity relationship. Okay, so here we tried to find out at what concentration of our proposed lead molecule or the ligand molecule it will act against the viral protease. Okay, so using BioVia Discovery Studio, we tried to find out different physicochemical properties of already reported inhibitors of those proteases, viral proteases. There are some already reported inhibitors which are published by different authors around the globe, and these data are available in the different literature. So we collected those data, and their inhibitory concentration were also there. So we tried to calculate the physicochemical properties for those already reported inhibitors, and we tried to establish a statistical correlation between the physicochemical properties of those already reported molecules with their bioactivity, that is the inhibitory concentration, at what concentration they are active against the viral protease. And we validate our equation using some already known set of molecules by putting them again in the equation, QSAR equation in the QSAR model, and we could predict a very good QSAR model which we published. So QSAR is an analysis where we try to predict the activity of docking. Will tell you that this is the amount of energy, this is the affinity of this molecule towards the target protein. But QSAR analysis can also tell you that at what concentration it may inhibit that particular target protein. So here these are different docking studies. The interactions can be seen with different. This is in the center. This is the ligand, and these are the active site amino acids. You can see and how it binds. And here you can see the QSAR model also. So this is the QSAR plot where we can see for the already known set of already reported molecules, their experimental activity versus predicted activity we plotted, and we got a very good plot, and we generated this QSAR equation. So log IC50 inverse is equal to this. This equation is there in this particular paper. Molecular weight, you will have to put number of rotatable bonds, then charm energy, total energy. So you will have to calculate. Suppose now you are going to study, you are going to propose a new molecule for as the SARS-CoV-2 protease inhibitor. So you have performed docking. Now, by calculating this, its molecular weight for your molecule, number of rotatable bonds, there are different softwares. You can calculate those these properties, and you can put those values in this equation. You will be able to calculate the log IC50 value for your molecule. So this is how QSAR helps us in predicting the bioactivity of unknown molecules. So with this, I will conclude my lecture, and my contact details are there in this QR code. So if you are interested, you can scan and you can contact me for any queries. We are also still learning these different tools. So if you have any questions, you can ask. I thank the organizers for giving me the opportunity to deliver my experience with the participants. Thank you.
Thank you so much, Dr. Chetia, sir, for clearly elaborating on the identification of disease targets and molecular interactions. The session was intriguing, at the same time extremely insightful, and the chat box reflects that the attendees have thoroughly enjoyed it. We shall now take queries.